The history of neurodegenerative disease research spans several centuries, but significant advancement made in the last few decades. In this video, we will explore the evolution of how our understanding of these complex disorders from the early observation to modern molecular and genetic insights. The earliest description of conditions resembling neurodegenerative diseases date back to ancient time. Chinese medical sources from around 425 BC and traditional Indian texts from approximately 1000 BC describe conditions that resemble Parkinson's disease, that is also known as PD. However, it wasn't until the 19th century that formal medical description of these disorders began to emerge. In 1817, James Parkinson published quote-unquote an essay on shaking palsy, which provides the first detailed medical description of condition now known as Parkinson's disease. This seminal work laid the foundation for the future research into movement disorders and neurodegeneration. Amyotrophic lateral sclerosis, that is also known as ALS, was first described in the 1869 by James Martin Charcot, a French neurologist. Charcot detailed clinical and the pathological observation of this motor neuron disease significantly advanced the field of neurobiology. In 1906, Alois Alzheimer presented the case of Auguste Dieter, a patient with what would later be known as Alzheimer's disease, AD. So Alzheimer's description of the characteristic plaque and tangle in the brain marked crucial moment in the history of dementia research. In early 20th century, research on neurodegenerative diseases primarily focused on pathological studies of post-mortem human brains, and these studies relied heavily on a standard histological technique such as hematoxylin and eosin, that is also known as HE staining, which stain protein pink and nucleotide purple respectively, as well as silver staining that highlight the protein and DNA in black in color. During this period, neuroanatomy was the prominent area of neuroscience. Neuroanatomists conduct detailed examination of neurons, glia, myelin, and synapse structure at both light and electron microscopic level. However, these tools for studying the living brain were limited, and the neurochemical study were also limited. The mid-20th century saw significant advancement in our understanding of the brain chemistry and their function. The discovery of neurotransmitters and their role in neuronal communication opened a new avenue for research into neurodegenerative diseases. In the 1960s, the dopamine hypothesis of Parkinson's disease emerged, leading to development of levodopa as a treatment. This breakthrough marked as a significant shift in the approach to neurodegenerative diseases, highlighting the potential for targeted therapy based on neurochemical imbalance. The 1970s brought about a revolution in neuroscience research with the introduction of immunohistochemistry techniques for the central nervous system, and this method allowed for more precise identification and localization of a specific protein in the brain tissue. In 1975, the presence of immune-related protein in the senile plaque of Alzheimer's disease patient was first reported, and this discovery hinted at the potential role of inflammation in neurodegenerative process, although at the time it was considered a secondary phenomenon rather than a primary causes. The 1980s saw the identification of microglial activity as a feature of neurodegenerative diseases. This finding would later prove crucial in understanding the role of neuroinflammation in this disorder. The 1990 marked as a turning point in the neurodegenerative diseases research with significant genetic discovery. In 1993, the Huntington gene responsible for Huntington disease was identified, and this was followed by discovery of genetic mutation associated with family form of Alzheimer's disease, Parkinson's disease, and other neurodegenerative disorders. During this decade, the role of neuroinflammation in neurodegenerative diseases began to gain more attention. A study found that the long-term user of non-steroid anti-inflammatory drug (NSAIDs) had a reduced risk of developing Alzheimer's disease, and this observation led to re-evaluation of the role of inflammation in neurodegeneration. So numerous papers elucidating the origin, nature and toxicity of microglia as well as their relationship with neurodegenerative diseases, which were published during this period. And these research efforts contributed to the growing recognition that neuroinflammation may play central role in the development of neurodegenerative diseases. As we enter in the 21st century, research on neurodegenerative diseases increasingly focus on molecular mechanism and protein aggregation. The role of misfolded protein in the various disorders became a central theme in the neurodegenerative disease research. For Alzheimer's diseases, the amyloid cascade hypothesis gained prominence, proposing that the accumulation of amyloid beta peptides was primary driver for the disease. In Parkinson's disease, the role of alpha-synuclein aggregation became a key area of investigation. So advanced imaging techniques such as positron emission tomography, that is PET, and the functional magnetic resonance imaging fMRI allowed researchers to study the brain changes in the living patient, providing new insight into disease progression and potential biomarkers. In recent years, the field of neurodegenerative disease research has experienced rapid advancement driven by the technological innovation and interdisciplinary approaches. Like genome-wide association study have identified the numerous genetic risk factors for various neurodegenerative. These studies have highlighted the complex genetic architecture of this disorder and the importance of gene environment. Next is epigenetic research has revealed the role of non-genetic factor in the disease development and their progression. A study has shown how environmental factors and lifestyle choices can influence gene expression and contribute to neurodegeneration. Next, the system biology approaches have allowed researchers to study neurodegenerative diseases from a more holistic perspective, considering the complex interaction between gene, protein and cellular pathways. So today neurodegenerative disease research is a vibrant and the multidisciplinary field, and the most studied neurodegenerative diseases include Parkinson's disease, Alzheimer's disease, ALS disease and Huntington disease. So recent research has increasingly focused on the role of neuroinflammation in the development and the progression of neurodegenerative diseases. There is a mounting evidence of the importance of immune responses in the neurodegeneration, with growing number of immune-related genetic mutations being identified as a risk factor. Researchers are also exploring the potential of stem cell therapy, gene therapy and immunotherapy for the treating neurodegenerative diseases. And these cutting-edge approaches offer hope for the developing more effective treatment in the future. So despite significant progress, many challenges remain in the field of neurodegenerative disease research. The complex nature of this disorder, involving multiple cellular and molecular pathways, make them difficult to study and treat. One major challenge is the lacking of effective treatment that can do more than just alleviate symptoms. While the drug like acetylcholinesterase inhibitor for AD and levodopa for the PD provide symptomatic relief. No current treatment can halt or reverse disease progression. Another significant challenge is understanding and addressing the causes of sporadic cases, which account for the vast majority of neurodegenerative disease cases. Unlike the familial cases with the clear genetic causes, sporadic cases often have no obvious underlying causes beyond age as a risk factor. So looking to the future, researchers aim to develop therapy that can delay the onset and even reverse the symptom of neurodegenerative diseases. And the goal is to create personalized treatment based on genetic and biochemical study. Biomarkers such as imaging technique and the blood-based tests, proteomics and metabolomics, are being developed as a screening tools and the predictor for disease progression. And these tools could allow for earlier diagnosis and intervention, potentially improving patients' outcomes. So now I would like to conclude here that the history of neurodegenerative disease research is a testament to the power of scientific inquiry and an interdisciplinary collaboration. From early clinical description to modern molecular and genetic insight, our understanding of these complex disorders has grown tremendously. While significant challenges remain, the rapid pace of scientific advancement offers hope for the future. As we continue to unravel this complexity of the neurodegenerative diseases, we move closer to developing effective treatments and ultimately cure for these devastating disorder. So the journey from the James Parkinson essay on the sacking palsy to today's advanced genetic and the molecular study illustrate the remarkable progress made in just 2 centuries. As we look the future, it's clear that the field of neurodegenerative disease research will continue to evolve, driven by the technological innovation, interdisciplinary approaches, and the urgent need to address these increasingly prevalent disorders in our aging global population. So thank you for the watching and see you in the next video.